Hello, we will be talking about antimicrobial peptide membrane interactions in this webcast with Dr. Liqun Zhang from the Tennessee Tech University. Dr. Zhang, thank you so much for taking the time today.
Oh, my pleasure. Thank you for giving me a chance.
Dr. Zhang, we will be talking about your paper published this year in the journal ACS Omega. Can you maybe first tell us about the key findings of your paper? And also, did you expect those results when you started your investigation?
Sure. So we published a paper on ACS Omega. In the published work, we did molecular dynamic simulations to understand the structure, dynamics, and translocation capability of human beta-dependent type 3 through different membranes in various oligomerization forms. Human beta-dependent type 3, abbreviated as HBD3, is a small cationic cysteine-rigid peptide belonging to the human innate immune system. It has a broad spectrum of antimicrobial activities, but at high concentrations, it also shows hemolytic activity by interrupting red blood cells. In order to understand the selectivity of HBD3 disrupting different cell membranes, investigating the capability of HBD3 translocating different membranes is important. And we did both all-atom and cross-grained molecular dynamic simulations on HBD3 in monomer, dimer, and tetramer forms, translocating through the model red blood cell membrane and photobancterial membranes. And we found that membrane thinning is a common phenomenon around HBD3 in different kinds of lipid membranes and in different HBD3 oligomer systems. However, if the molecular structure is more rigid, then this kind of membrane thinning will be more significant. We also found the rotation of the oligomer inside the bilayer and the stretching of the oligomer structure along the membrane, normal direction. So also calculating the free energy of HBD3 through different lipid membranes, we found that generally HBD3 encounters a lower energy barrier through different lipid membranes and through the negative charged membranes, then through the ionic membranes. And HBD3 in different forms need to overcome a positive energy barrier through the red blood cell membrane, and HBD3 tetramer needs to overcome a much lower energy barrier through the bacterial membranes. Okay, and also in a combined bacterial membrane, PC plus POPS bilayer, and if the HBD3 cross through the POPS leaflet, then the energy barrier is much lower than through the POPC leaflet. Also, the potential of mean force results suggest that HBD3 should form an oligomer for translocating through the negatively charged lipid membrane at a low concentration. So that's our findings.
And in your opinion, what could be maybe the further steps to promote the antibacterial effects and maybe also to lessen the side effects?
Yeah, that's a good question. So antimicrobial peptides such as defensin, they have low or no drug resistance because they kill bacteria by directly interacting with the bacterial membrane. Okay, because defensins are strongly positive charged with the charge density in the range of positive 3 to positive 11, they can interact with the negative charged lipid head groups in bacterial lipid membrane. Usually, the bacterial cell membrane is negative charged, or while the normal cells are overall neutrally charged, or with the negative charged lipid phosphotidylserine on the linear side of the cell. So HBD3 can distinguish between bacteria and normal cells and disrupt the lipid membrane of bacterial cell only. Cause cell leakage, thus to kill a microorganism. Because defensin kills a microorganism by disrupting the cell membrane, which is relatively stable in chemical composition and topology. So the defensin peptide-based antibiotics are unlikely to cause drug resistance. It has been found that the antibacterial activity of HBD3 is determined by its charge and also its structure. The analog form of HBD3 has been found to have a similar or slightly higher antibacterial activity than is wild type, based on our simulation work and also from other researchers' experimental work. In order to kill bacteria efficiently, the next thing what we can do is to increase the charge density. So in order to increase charge density, we need to have high enough concentration of HBD3. Because at a high concentration, HBD3 can crowd and aggregate on the membrane and then cross the membrane in an oligomer form. However, if the concentration of HBD3 is too high, it can cause hemolytic effect by disrupting normal red blood cell membrane. So finding out the critical concentration of HBD3 is important, which can help to promote the antibacterial effects and lessen the side effects. Okay, and in general, we know that drug resistance is one of the biggest public health challenges or side effects of antibiotics of our times. Each year in the United States, at least 2.8 million antibiotic-resistant infections occur, and more than 35,000 people die as a result, according to CDC's 2019 antibiotic resistance threat report. Okay. The future of discovering no generation of drugs with no or low drug resistance to treat bacterial infection relies heavily on the continuous acquisition of knowledge and discovery of new groups of antibiotic peptides. At this moment, the main barrier in designing novel drugs lies in not enough understanding of AMP's antibacterial mechanism in molecular detail. For example, the antibacterial functional mechanism of defense, such as HBD3 in molecular detail, is still unknown. Okay, so more molecular level research should be conducted to understand the functional mechanism of defense in the future, and based on which novel drugs should be designed to promote the antibacterial effects and lessen the side effects of antimicrobial peptides in the long run.
Are there other known substances that use the same way to get into the cell? And if so, what do they look like, or what could they be used for?
Yeah, that's a good question. Yes, there are other antimicrobial peptides, which also can directly disrupt the bacterial cell membrane, break the membrane just to kill the bacteria. So there are three prevalent theories of the membrane disruption mechanism. First one is the carpet model. Okay, in the carpet model, the antimicrobial peptides cover the membrane surface before dissolving the membrane like detergent, leading to the local disintegration, including the membrane methylation or fragmentation upon reaching a critical concentration. The second is the pore formation model, which can be further classified into the barrel stuff model and the toroidal model. And in both, the AMPs form a pore inside the membrane. So if the lipid has a significantly reoriented to the protein, and all the lipid monolayer bend continuously through the pore, then with the polar defense phases associating with polar lipid head group, then it should be the toroidal model. Otherwise, it is the barrel stuff model, in which the hydrophobic peptide regions align with the lipid core, and the hydrophilic peptide region form the interior of the core. So those are three prevalent theories. And there are other antimicrobial peptides. They just also can disrupt with the bacterial membrane directly. For example, it was found that antimicrobial peptide called alamethine in hexamer form can form a barrel stuff pore in membranes and cause cell leakage and kill the cell. Okay, and the melatonin in a tetramer form can form a toroidal pore and can kill the bacteria. Okay, and the plant defense named NAD1 can exploit membrane phospholipids to induce cytolysis in target cells. It forms a complex comprising 7 NAD1 dimers and 14 PIP2 lipids, which is a process associated with formation of membrane blebs and membrane rupture, thus causing cell lysis.
What would the further research in this area look like? Maybe you can share with us your opinions on the next steps in this research.
So in the future, more detailed information on structure and dynamics of defense translocating through explicit models of gram-positive and gram-negative vector membranes should be conducted, with the goal to find out the antibacterial mechanism of defensin in molecular details and to design novel drugs. Okay. Besides having a broad spectrum of antibacterial activities, defensin also has microbicidal activity against the fungi, yeast, and some enveloped viruses. Recently, it has also been found that defensin, such as human beta defensin type 2, human alpha defensin type 5, can block the COVID-19 virus from binding with ACE2 receptor on normal human cells. Okay, so more researches should be conducted to understand the functional mechanism of defensin interacting with bacteria and with viruses in more molecular detail. And in order to find out the clues to solve drug-resistant issues and to combat COVID-19 virus and its adverse variants at the end.
Dr. Zhang, again, my thanks to you for participating in this webcast. I wish you a very enjoyable holiday season and continued success with your work in the new year. Thank you.
Thank you for giving me the chance to talk about my research, and really a pleasure talking with you and talking about the work, and also have a great holiday season to you and enjoy.
Thank you. Thank you, Dr. Zhang.